In this video, we're going to look at metabolic traffic control. So how is it that cells regulate how much glucose enters into glycolysis and how much product is being made? In a previous video, I gave you this broad overview of metabolism in an animal cell, and I expect you to remember that glucose can be broken down to form pyruvate through glycolysis, and then that pyruvate can either enter the fermentation pathway, or it can be metabolized further through the TCA in mitochondria. In this video, we're just focusing on glycolysis and how that flow gets regulated. Well, I hope you watched that video and I hope you took notes and that they look something like this. Just as a reminder, in a cartoon like this, each one of the circles represents a carbon, so we're following the reaction by looking at where the carbon and the phosphates are. Here's a more standard way of looking at glycolysis in which the different intermediates are drawn out. And what I want you to notice is that glycolysis breaks down glucose to yield 2 pyruvate, 2 ATP molecules, and 2 NADH molecules. Now, all of the reactions in glycolysis are important, but in this video, we're going to focus on the four that get regulated quite a bit. These reactions will be the one that catalyzes step 1, that's carried out by hexokinase, step 3, that's carried out by phosphofructokinase, the last step, step 10, that's catalyzed by the enzyme pyruvate kinase, and then this step here, which is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. So these are our focus enzymes: hexokinase, phosphofructokinase, pyruvate kinase, and glyceraldehyde 3-phosphate dehydrogenase. And what I want you to think about is this. 3 of these enzymes include the name kinase in their name. So just in general, what is a kinase? Well, the answer is that a kinase is the enzyme that catalyzes the addition of a phosphate group to its substrate. Let's look at these enzymes now, beginning with hexokinase, which catalyzes the first step in glycolysis. Well, here I'm showing you the substrate glucose and the product, glucose 6-phosphate, both as their actual chemical structures, but also with the dots that represent carbons. And the first thing that I want to do is show you that this enzyme really is catalyzing the addition of a phosphate group. So you can see the phosphate on the 6 carbon in glucose, and you can see that that phosphate is being donated by an ATP. So the enzyme hexokinase is catalyzing phosphorylation of the substrate. Now let's look at regulation. So the main way that the hexokinase enzyme is regulated is when there's high amounts of glucose 6-phosphate, it will inhibit the hexokinase enzyme. This is an example of something we call product inhibition, and that's just the general name that we give to a type of inhibition where the product of the enzyme, in this case glucose 6-phosphate, binds to the enzyme and inhibits its activity. Now, products generally resemble the substrate, and this type of regulation is usually competitive. Well, this gives me a chance to quiz you about enzyme kinetics. So suppose that we measured the kinetics of the hexokinase enzyme with and without glucose 6-phosphate. Which of the following would be different? Would the Vmax change? The Km? Neither? Or both? Well, if glucose 6-phosphate's a competitive inhibitor of hexokinase, we'd expect Vmax not to change, but Km would increase. And in fact, that is what you see if you do experiments on hexokinase. Let's look at our second focus enzyme. This is pyruvate kinase, which catalyzes the last step in glycolysis, step 10. Now, this is the reaction that takes phosphoenol pyruvate and then converts it into pyruvate, generating ATP. I've added the structures of phosphoenol pyruvate and pyruvate so you can see them. And one thing I want to point out, I colored the carbons in with black dots for phosphoenol pyruvate because I wanted to convince you that if you look at it, you really can see that there's a phosphate on the 2 carbon. You could recognize this compound. Now, you might be wondering why this enzyme is called pyruvate kinase when the substrate is phosphoenol pyruvate, because enzymes are normally named after their substrate. The answer is that this enzyme was first discovered by biochemists studying the reverse reaction, so pyruvate reacting with ATP in order to produce phosphoenol pyruvate and ADP. In the cells, it's normally running in the direction that I've drawn it, with phosphoenol pyruvate as the substrate. Well, let me take these away so we can talk about regulation now. Well, it turns out that pyruvate kinase is regulated by a lot of different factors. ATP and alanine both inhibit it. Fructose 1,6-bisphosphate activates it. Let's look at these and see why that might be. I'm going to begin with ATP. Well, this is an example of product inhibition, right? ATP is a product of the reaction, and so it's inhibiting the pyruvate kinase enzyme. That should make sense. What about alanine? Well, this is a little trickier because I have to talk about alanine in the cell. So you should know that pyruvate is a major branch point in the cell. It can feed into fermentation or the tricarboxylic acid cycle, but pyruvate can also be converted into the amino acid alanine. So what we're seeing here is an example of something called feedback inhibition. And that's when a compound in a metabolic pathway, so this would be how are we going to synthesize alanine? What's the metabolic pathway for that? And that compound feeds back onto an earlier step and inhibits the enzyme catalyzing that. So it's not product inhibition because we have to go more than one step back. And in feedback inhibition, we generally don't have competitive inhibition because the product looks different from the substrate, and so it's binding to an allosteric site. This would be an example of non-competitive inhibition. Okay, well, what about fructose 1,6-bisphosphate? That's acting as an activator. To explain this, I think it's useful to think about the broader glycolysis pathway. Here's our focus enzyme, pyruvate kinase, and back here is fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate is a compound that shows up earlier in this metabolic pathway. Fructose 1,6-bisphosphate's action on pyruvate kinase is an example of feed-forward activation. And basically that's when an intermediate in a metabolic pathway feeds forward and then speeds up the activity of an enzyme catalyzing the later step in that pathway. And again, this would act by binding some allosteric site in the regulatory domain. Now there's one more way that pyruvate kinase is regulated. The way that I've drawn pyruvate kinase here, it's on, right? It's obviously has high activity because we're able to catalyze this reaction. But pyruvate kinase is one of those enzymes that can switch between an on state and an off state. And this will depend on whether or not it's phosphorylated. So if a phosphate group is added to pyruvate kinase, it goes off. It has this low activity and not much product will be made. This is an example then of a process that we call covalent modification. So that's when an enzyme can switch between a high activity and a low activity state with the addition or removal of a phosphate group. And in this particular case, what we're seeing is that phosphorylation turns it off. So there's an enzyme called protein kinase A, which can phosphorylate pyruvate kinase, turning it off. There's another enzyme called phosphoprotein phosphatase 1, which can remove that phosphate group and turn it back on. So this is one more way that cells can regulate the flow through glycolysis. I've saved this step for last. This is the third step in glycolysis. It's catalyzed by the enzyme phosphofructokinase. And what I want you to know is that this is the most regulated step in glycolysis. And you might wonder, well, why? And the answer is going to be, it represents a commitment step. And what that means is that once the cell runs this reaction, the product, this fructose 1,6-bisphosphate, has no alternative but to follow through the rest of the reaction. And while there are earlier steps in glycolysis, the products of those have more than one possible fate. So fructose 6-phosphate could be converted into fructose 1,6-bisphosphate, but it might also be diverted and used in different pathways. But if the cell runs phosphofructokinase, it's committed to carrying that out until we get the pyruvates at the end. There are many, many things that regulate phosphofructokinase. It can be activated by AMP and by fructose 2,6-bisphosphate. It can also be inhibited by ATP, by citrate, that's a compound found in the tricarboxylic acid cycle, and by phosphoenol pyruvate. Now, do you notice anything odd about this list of inhibitors? Well, what I'm hoping that you're going to notice is that it's a little weird that this enzyme is inhibited by ATP, because ATP is actually a substrate for phosphofructokinase. So how can it be that a single molecule can be both a substrate and an inhibitor? Well, when this happens, we say that it's an example of substrate inhibition. So if there's a high concentration of the substrate around, in this case ATP, it inhibits the reaction that would consume it. This isn't very common. It's not common in metabolism, but it's not unique to PFK. Another example is that high concentrations of pyruvate inhibit LDH, the enzyme that catalyzes lactate fermentation. Okay, well why? So I'm going to move this aside so I've got a little bit more room, and let's think about this. So suppose that this is our phosphofructokinase, and now I've drawn it a little bit differently so that I can show you that, like most enzymes, there's a catalytic domain and then there's a regulatory domain. And if there's very little ATP around, if there's only a low amount of ATP around, it'll bind with a high affinity at the active site, and then we'll run this reaction. But suppose that there was a lot of ATP present in the cell. Well, under those circumstances, the active site might already be filled with an ATP molecule, so there's another lower affinity site, and ATP can bind to that on the regulatory domain, and when it does this, it puts the brakes on the reaction. For phosphofructokinase, the reason that we would do this is if the cell has lots and lots of ATP, there's no point in running glycolysis because glycolysis products can feed into the TCA cycle. And instead we could do things like take the glucose or the fructose 6-phosphate and use them elsewhere in the cell. All right, let's recap. We've looked at hexokinase, phosphofructokinase, and pyruvate kinase. The last of our focus enzymes is glyceraldehyde 3-phosphate dehydrogenase. Okay. Let me begin by saying that when you see the name dehydrogenase enzymes, those will catalyze oxidation reduction reactions. So that's a clue for you. Now, I assume that you've had chemistry and you know about redox reactions from chemistry. There's always an electron donor and then an electron acceptor. And what happens in a redox reaction is the electron donor donates an electron, so it becomes oxidized. And then the electron acceptor accepts that electron, and we say that this is being reduced. So you lose an electron, you're oxidized. You gain an electron, you're reduced. Here's my tip for you. In biological redox reactions, the movement of electrons is often accompanied by a proton. And so when we look at those reactions in metabolism, it looks like we're adding or removing a hydrogen. Let's see if this holds up when we look at glycolysis step 6, which is catalyzed by a dehydrogenase. All right, well, it's a little hard to see hydrogens showing up and disappearing here because I've drawn it with my carbon balls and phosphates. But here's an image of what the reaction looks like. So on the left, we have glyceraldehyde 3-phosphate, and then there's an inorganic phosphate, and that will then combine to make 1,3-bisphosphoglycerate, while at the same time taking NAD+, this molecule, and converting it into NADH and a free proton. Well, let's test what I just told you about hydrogens appearing and disappearing. So go over here and look at the substrates and the products. See how in glyceraldehyde 3-phosphate we've got these hydrogens? And then if you look at glyceraldehyde bisphosphoglycerate, those hydrogens are gone. And if we look at NAD+, and then we look at NADH, you can see that the hydrogens appear to have been transferred so that we've got NADH and a free proton. I can show you this in a little bit more detail when we look at NADH and NAD+. NAD stands for nicotinamide adenine dinucleotide, and if it's in the oxidized form, we say it's NAD+, and if it's in the reduced form, we say NADH. And here are the structures. Well, this is a chance for another pop quiz. Nucleotides are made out of a phosphate group, a ribose, and then a nitrogen-containing base. Can you identify those components in NADH or NAD+? You should be able to. So here, what I've just circled is the nitrogen-containing base adenine, sugar ribose, and a phosphate. Over here, there's a different nitrogen-containing base, that's nicotinamide. There's a ribose and a phosphate. So together we're going to call this nicotinamide adenine dinucleotide, two nucleotides that have been joined together. For this particular reaction, we're looking at the redox reactions. And so the next question would be, well, can you see the gain or loss of a hydrogen? And if you study this for a little bit, you'll say, yeah, I can. Here on the NADH, we can see the addition of a hydrogen. It's absent in the NAD+. I also want to point out that NAD+ has picked up an electron because it's gone from being positively charged to being neutral in NADH. Glyceraldehyde 3-phosphate dehydrogenase is the only dehydrogenase in the glycolysis pathway. But as we go on and we look at other pathways, both fermentation and reactions in the TCA cycle, we'll see many more dehydrogenases, and it will really help if you can remember that what we're doing is we're studying redox reactions and look for that appearance and disappearance of a hydrogen.